Hello everyone, today we will continue with cell division, specifically looking at meiosis. This process is used for gametogenesis, which means that it is used to turn diploid cells, which are spermatogonia and oogonia, into haploid cells. There are two main stages, meiosis 1 and meiosis 2. Each of these stages have phases that resemble those phases of mitosis. By this, I mean that meiosis 1 would have interphase 1, prophase 1, metaphase 1, and so on, and so would meiosis 2. Now let's first of all start with meiosis 1. Meiosis 1 begins with interphase 1, which is basically when the DNA replicates itself. As you can see here, these two are homologous chromosomes, and so are these two. When they replicate, they look like this. Prophase 1 is when the chromatid loops coil up and then they condense to take up that shape of a chromosome, and then the homologous chromosomes will begin to line up side by side like this. Of course, after that, the nuclear membrane disappears, therefore the chromosomes are free in the cell like this. As well as that, the spindle fibers also begin to form. It is important to consider that although the homologous chromosomes line up side by side like this, sex chromosomes in males, i.e. the X and Y chromosomes, they do not line up like this. They line up end to end because they are not technically homologous. As you can see here also, where the chromosomes cross each other, it is called a chiasma, and at this chiasma is where the genetic material exchange takes place, which gives a lot of variation. By the process of synapsis, the homologous chromosomes pair up side by side, like I've said, and also the chromatids of homologs intertwine, and then they become what is called bivalent or tetrad. Bivalent refers to the fact that there are two chromosomes together, or tetrad refers to the fact that there are four chromatids. Prophase 1 is a very important phase in meiosis, and it distinguishes meiosis from mitosis. Next, we have metaphase 1, which is basically just when the homologous chromosomes line up at the equatorial plane of the cell due to the spindle fibers contracting a little and pulling them into place. Afterwards, we have the anaphase 1, which is basically when the chromosomes are being pulled apart to the opposite poles of the cell. You will notice here that the chromosomes have exchanged their genetic material. So the homologous chromosomes will contain bits of the other homologous chromosome, as you can see here. Also, chromosomes migrating to each pole will contain one member of each autosome and one sex chromosome. Telophase 1 then occurs when the two new sets of chromosomes have migrated to opposite poles of the cell, and then a new nuclear membrane has formed around each new set of chromosome. The chromosomes would have loosened up a little bit at this point as well. Cytokinesis then occurs in humans, and in males, the cytoplasm would be divided about equally between the two daughter cells. However, in females, most of the cytoplasm will go to one cell, and as a result, for males, there would be two even and functional daughter cells produced. However, for females, only one functional cell is produced, and the other cell would be a polar body. Meiosis 2 then begins when interphase 2 begins, and it is notable that in this phase, the DNA is not replicated, unlike in interphase 1. Next, we have prophase 2, which is very similar to that of mitosis. The chromatin loops basically condense, the nuclear membranes disappear, and the spindle fibers begin to form. Next, then we have metaphase 2, which is just basically when the spindle fibers form across the cell like this, and the chromosomes are positioned at the equatorial plane of the cell. However, you need to note that in metaphase 1, the spindle fibers are formed in this direction, whereas in metaphase 2, the spindle fibers are formed in this direction, so the directions are perpendicular to each other. Next up, we have anaphase 2, which is basically just when the chromosome is pulled apart at the centromere, so each chromatid is pulled towards the pole of the cell. Once again, once they are pulled apart, this is not called a chromatid anymore, and it is called a chromosome. Telophase 2 then begins when the chromosomes have migrated to opposite poles of the cell and a new nuclear membrane have formed around each new set of chromosome, and the chromosomes would once again have loosened up a little bit at this point. It is once again notable that although cytokinesis happens in all humans, the cytoplasm divides evenly for only male gametes. So you would end up with 4 functional and even gametes for males. However, in females, once again, most of the cytoplasm goes to only 1 cell. So you would get only 1 functional cell that can actually be fertilized. And then another polar body, as mentioned before. However, the polar body that was made in meiosis 1 may also undergo meiosis 2. And if that happens, you would end up with 1 functional egg and 3 polar bodies that would eventually degenerate. Although this is labeled final products, like I've said before, this would only be true for males. And however, for females, there would be a really big gamete and 3 polar bodies that are basically useless. Thank you so much for watching. Please like and subscribe for more.